Bismillahirrahmanirrahim. Assalamu alaikum dear students. Today's our lecture is about the heteropolysaccharides. As it is shown by the names, the heteropolysaccharides are those polysaccharides which contains which upon their hydrolysis give rise to the different monosaccharides, more than one types of monosaccharides. And also the non-carbohydrate parts are also incorporated in the heteropolysaccharides. Heteropolysaccharides are also classified into two types. One are the plant heteropolysaccharides and the second one are the animal heteropolysaccharides. Heteropolysaccharides. As you studied earlier, the homopolysaccharides in which the polysaccharides only yields upon complete hydrolysis only the one type of monosaccharidal units. But in case of heteropolysaccharides, they yield different types of the monosaccharidal units upon complete hydrolysis. They are of two different origins, plant and animal. Depending upon their source, they are classified into plant heteropolysaccharides and the animal heteropolysaccharides. In case of plant heteropolysaccharides, which are also called as the mucilages. The second name of the plant heteropolysaccharides are mucilages. The animal heteropolysaccharides are also called as mucopolysaccharides. The types of the plant heteropolysaccharides are agar, pectin, and chitin. Their types are hyaluronic acid, Their types are hyaluronic acid, heparin, chondroitin, sulfate, and the blood group substances. Also, the last one are the bacterial polysaccharides. From the animal origin, the mucopolysaccharides are subclassified into the different forms: hyaluronic acid, heparin, chondroitin sulfate, blood group substances, and the bacterial polysaccharides. In case of plant, they are called as the mucilages, and their different types are the agar, pectin, chitin. Now we will discuss them one by one. First one is the agar. It is present in seaweeds, upon hydrolysis it yields different types of monosaccharide units and also non-carbohydrate compounds like sulfate and phosphatases. Heteropolysaccharides upon hydrolysis give rise to the different monosaccharidal units and also non-carbohydrate compounds like the sulfates, phosphates, etc. In case of agar, agar is present in high amounts in the seaweeds. The source of the agar is seaweeds and it is the polymer of sulfated galactose. Polymer of sulfated galactose. It is a polymer of sulfated galactose units present in it. Agar is dissolved in hot water and sets to a gel on cooling. So that's it. Due to this property of the agar, this one is a very much rich source for the cultured medium of the different types of bacteria, and also it is used for the polyacrylamide. It is also used for the formation of gels for the identification of the DNA and RNA molecules in case of the agarose gel. For the formation of agarose gel for identifications of nucleic acids. Also, agar is used for the treatment of the constipation because it is not utilized by the humans and adds to the bulk of the feces and helps in its propulations. So it is very helpful for the get rid of the constipation, for the treatment of constipation. Agar is used as culture media for bacterial strains and also used for the formation of agarose gel. Next one after the agar is the pectin. These are present in the apple, lemon and other fruits. It also forms gels with sugar solutions. They are the polymers of galactonic acid galactose, and the pentose sugar, arabinose. Pectin is the polymer of galactonic acid, galactose, and arabinose, the pentose sugar. Next one is chitin. This is present in the exoskeleton of invertebrates like carps, lobster, and insects. Chitin is actually the exoskeleton of invertebrates like crabs, lobster, and insects. It is made up of acetylated glucosamine units. This is actually the polymer of acetylated glucosamine units. These all are the plant heteropolysaccharides. Now we going towards the animal heteropolysaccharides. These are present. The animal heteropolysaccharides are usually present in combination with the proteins and as the mucoproteins. They are called as the mucoproteins. Hyaluronic acid, heparin, chondroitin sulfate, blood group substances, and the bacterial polysaccharides belonging to this category. First one, we will discuss the hyaluronic acid. Hyaluronic acid, the first one, it is present in the synovial fluid, skin, and in some microorganisms. It consists of acetylated glucosamine, and glucuronic acid units. This is present in the synovial fluid, skin, and in some microorganisms, and it is the polymer of the acetylated glucosamine and glucuronic acid units. In a highly polymerized state, it acts as a lubricant and the cementing material. It acts as It acts as the cementing material and lubricant in cells, between the cells. It acts as the cementing like the cementing material and the lubricant in the cells. In the cells, in case of the cells, in certain bacterial products, the hyaluronidase enzyme is present that break down this hyaluronic acid and make the tissues readily permeable to the bacteria and their toxins. That's why these hyaluronic acid acts as the lubricating and cementing material between the cells. In the absence of this lubricant material, the bacterial cells can easily be penetrating into the cells and damages the cells by their toxins. Next one is the heparin. What is the role of the heparin? Heparin. It is an anticoagulant agent substance produced by the mast cells. Heparin. Heparin is the anticoagulant substance produced by the mast cell. It is the polymer of glucosamine and glucuronic acid. It is a polymer of glucosamine and glucuronic acid units. These units are sulfated rather than acetylated. Heparin acts as the anticoagulant substance that is produced by the mast cells. It is the polymer of glucosamine and glucuronic acid, sulfated rather than the acetylated units. Next one is the chondroitin sulfate. Next one is chondroitin sulfate. It is present in the ground tissues of in the ground substance of connective tissues and cartilage. It is present in the ground substance of the connective tissues and the cartilage. It is the polymer of acetylated galactosamine and glucuronic acid. Units: galactosamine also sulfated at 6 and 4 positions in some units. This is the polymer of acetylated galactosamine and glucuronic acid, and the galactosamine units are sulfated in some units at the 6 and 4 positions by the sulfuric acid. Next one is the blood group substances. The examples of heteropolysaccharides: blood group substances. These are the A, B, and RH antigens of erythrocytes These are the A, B and RH antigens of the erythrocytes that are the natural mucopolysaccharides. Combined with proteins these consist of glucosamine, galactosamine, polymers of the glucosamine, galactosamine and other sugar derivatives. Blood group substances, the antigens A, B and RH antigens of erythrocytes are actually the natural mucopolysaccharides combined with proteins. It consists of the glucosamine, galactosamine and the other sugar derivatives present in them. The next one are the bacterial polysaccharides. In some types of bacteria, some bacteria contains the special bacterial polysaccharides that are like the salicylic acid containing of salicylic acid. These are the special types of bacterial polysaccharides that are present in some types of bacteria. These are also the heteropolysaccharides. Heteropolysaccharides have also very much importance in case of different benefits achieved by these heteropolysaccharides. Heteropolysaccharides are very much important for the different functioning of the cells, as they are acted as the lubricants in case of the cells. They provide the lubricants, they provide them the strength, they are the anticoagulants, they are the ground substances of the connective tissues and the cartilages. They are helpful for the blood group transfusions because they are the blood group substances which give rise to the blood group systems. By different blood group systems, the blood groups of the persons can be identified by these identification of these blood group substances present in that blood. They are also important for the identification of the foreign particles invaders into and lure down the entry of these particles. And also they are important for the invertebrates because they form the exoskeletons of the different invertebrates like the crabs, lobsters and insects. And they are used for the treatment of the constipation. They are important for the formation of the different types of the fruits. They are present in the different types of fruits and give them the texture. And also they are important for the formation of the culture media for the different types of the bacteria to be cultured like the agar. And also they are important for the other benefits of the heteropolysaccharides. You have know about all the heteropolysaccharides that are very important parts of the polysaccharides. Now you have idea about the heteropolysaccharides. If you have questions regarding the heteropolysaccharides, you may ask me by putting the comments in the comment section of the YouTube channel. And also you can approach me on my email address by sending me an email on my email address. And also you can approach me on my WhatsApp group by knowing about the lectures. This course is about the different types of the biochemistry of the carbohydrates. You have studied different types of carbohydrates and now you are studying the polysaccharides. In case of polysaccharides, now the remaining the polysaccharides are completed. Now we will go towards the blood group substances where they have different blood group systems which are we have to discuss in the next lecture about the blood group systems. That is definitely related to the heteropolysaccharides because the blood group substances are the part of the heteropolysaccharides, and they are actually give rise to the blood group systems. Thank you so very much, students. That's all about today's lecture. If you have any questions regarding this lecture, you can approach me on my WhatsApp group. You can email me on my email address, or you can put in the comments in the comment section of the YouTube channel. I hope you understand the ideas about the heteropolysaccharides. Thank you so very much, all of you. Allah Hafiz.